All right. Excellent. Thank you very much, Nia, for technical skills. So I actually run a computational lab, and you would not know it, would you? All right. So as I mentioned, I do run a computational lab. I thought I'd just mention a little bit about the research. If any of you have anything you want to discuss with me, I'm more than happy to kind of discuss some of these kind of research ideas that we're doing. So I'm based at Sick Kids, and we do largely work on parasites and microbiome. So in terms of microbiome, we look at IBD, we look at malnutrition. So I mentioned yesterday that one of the studies I'm most excited about is this one looking at malnutrition during pregnancy. And this is through a collaboration with colleagues at the Aga Khan University in Pakistan. And they've recruited 400 patients. We also have a node at St. Michael's Hospital in Toronto. And over the same period, we've only been able to recruit 30 patients. So it really shows what a machine they've got working in at the Aga Khan in being able to recruit so many women into these kind of studies. We've been working on parasites since forever. And now we've been able to merge our interest in parasites with microbiome. So I mentioned yesterday that there's a lot of focus on bacteria in the microbiome, but there's other things in the microbiome as well. So there's fungi, and some of you are working on fungi. I'm particularly interested in protists, and these are relatively understudied. Every time we sequence a new person's microbiome, we always find protists. So whether they're in Canada, whether they're in Pakistan, whether they're in Malawi, we always find different sets of protists, and we have no idea if they're contributing to disease or not. So the idea that some of these are contributing to disease, some of them can cause gastrointestinal problems. So Giardia is one that we may be familiar with. Giardia can actually be a commensal organism. So a lot of people have asymptomatic infections with Giardia. And so we're interested in understanding what is the switch that causes some of these commensal organisms to shift to pathogenicity, so these so-called pathobionts. We work on gonorrhea. And again, we're trying to push that towards more of a microbiome focus as well, and see how the microbiome of the genital tract contributes or exacerbates to disease. And chickens. So I work in a children's hospital, and it amuses a lot of my colleagues that we do work on chickens. Turns out it's a lot easier to get funding to study chickens than it is to get funding to study microbiomes in children. And we've been using chickens now for about 10 years to develop a lot of the methods that we're now starting to apply to human microbiomes. So it's really proven very, very useful in terms of giving us a model to start exploring some of these kind of diet microbiome interactions and some of the methods that we need to develop. All right, so what are we doing today? Yes.
A quick question about your chickens.
Yes.
So you're using chickens as a model for human microbiome. How similar are they?
No. No. It's a model for developing the approaches. Okay. Yeah. So if something's going to work in chickens, you know that it's going to work in humans. But it is monogastric, right? So and there are different compartments, and there's different compartments, but the ceca, which is where most of the fermentation, that's a very different kind of tissue relative to anything that I guess our closest would be the appendix, but it's a major site within chickens, for example. So chickens, much more genetically homogeneous. The feed is very consistent as well, and so you're starting to minimize some of the variation that you have with human microbiomes, for example. Oh, okay. So in terms of what we're aiming to do, so this morning we're going to be focusing on annotating MAGs in terms of function, taxonomy, and this involves predicting gene models, so identifying genes within these MAGs. And then once you've identified these genes, how do you actually annotate them with some sort of function? And then there's this concept of these kind of more generic annotations, so these broad functional categories like gene ontology and COG and so forth. But how do we get more insights into more detailed kind of subsystems? And this is an area that we're particularly interested in. And so whenever we publish a new study on using a metatranscriptome, we try and bring in a new subsystem to kind of focus on. So we're building a repertoire of these different subsystems that can start informing on the different functions associated with the microbiomes that we study. I'm going to spend some time on metabolism. Metabolism is probably the best studied system. In terms of bacteria, probably about anything from a quarter to a third of all of the genes associated with bacteria are associated with metabolism. So you can learn a lot about function of bacteria just from looking at metabolism. Very well studied, very easy to characterize, very easy to understand what is going on using metabolism as a functional focus. And then I want to shift gears a little bit. I want to talk about metatranscriptomics, and a few of you are already interested in doing metatranscriptomics, and I wanted to go through what are some of the differences in metatranscriptomics relative to metagenomics, and try and convince you as to the benefits of doing metatranscriptomics. All right, so here is basically how we ended up yesterday. We assembled contigs, we generated our sets of mags, and so now we want to learn something about these mags. So what we need to do, we need to identify the genes, we need to identify the taxa that are associated with those mags, and we want to identify functions associated with the genes that are encoded in those mags as well. And so once we do this kind of annotation, then it sets us up for the more interesting analyses, the downstream analyses, where we can start doing comparisons across different communities and see what functions are changing in response to certain perturbations, environmental perturbations that we may be imposing on these microbiomes. Okay, so the basic concept in annotating these mags, now there's a number of different tools that are available to do this. So I think in 2012, PROCA was this automated tool that was applied to genomes. You can apply it to mags. That's been superseded, I think 2020, and there's a new release, BACTA 2 in 2024. So BACTA is the new kind of PROCA. So this is now the standard tool that we're using for annotating these mags, and BACTA will be part of the lab that you're going through. So the idea is, first of all, you need to identify the genes that are on your contig. Then once you've identified each of these genes, then you can use some sort of sequence similarity tool or some other type of annotation tool to actually assign some sort of function associated with each of those genes. And then some of these tools also enable you to assign taxonomy. One of these is the GTDB TK tool, a bit of a mouthful. So it's a similar kind of concept here. You're doing the gene calling with this tool. You're aligning this genome with all of these different marker genes to a reference database, and then you're using that to do annotation. So it's really focused on doing the annotation and coming up with potentially strain-specific information on each of these mags. So in terms of identifying genes, so many of these annotation tools rely on this algorithm called Prodigal, which has recently been updated to Pyrodical because it's written in Python. And when you look at a bacterial genome or part of a bacterial genome, it's actually quite easy to see these open reading frames. So here on the display below, we're effectively showing in blue these kind of potential open reading frames. So these are stretches between start codons and stop codons. And so they're quite easy to spot within bacteria, much harder for eukaryotes because you've got a lot more splicing and all these other kind of complications that are going on. But in bacteria, at least, it's pretty easy to identify these open reading frames. And then Prodigal or Pyrodigal also include GC biases and hexamer coding scores that can also identify which of these kind of open reading frames really encode for protein coding genes. Okay, so this is relatively straightforward, and I don't think very much has changed over the last 10 years or so. I think identifying genes is a relatively trivial exercise. I don't know if anyone else has any comments on that, if they haven't found that to be the case. Great. Okay, so you've got your list of genes. Next, what you want to do is to assign some kind of function. So there's a number of tools that are available to do this. The simplest are just these sequence similarity BLAST type matching tools, which look at sequence similarity. But there's other more sophisticated tools. So Hammer, for example, and the PFAM domain is a great way and very sensitive, very precise of identifying protein domains, which can give you ideas about function about a protein where there might not be a match to an existing protein. Yes.
Is it going off of like a known database of all genes, and like can you add genes that you're specifically looking for?
Absolutely. So I will get a little bit into this choice of database. So absolutely. So these kind of searches, you have your searching tool, and then you have your database, and the choice of database really is absolutely key. So if you're trying to identify protein functions, you can use RefSeq or UniRef, or there's this thing called all the bacteria, and that will serve as a database to try and find something that's already known out there, right? So that you want to ascribe something to each of your genes. So absolutely, you can feed in whatever databases you want. However, there are some caveats to that, and depending on the choice of database, you could end up with a lot of false positives, as we may see. Who has heard of annotation snowballs? Okay, well, let's go through annotation snowballs. So this is something to be aware when you're doing annotation. So in this particular example, here you've got protein B, which may not have a known function, right? And you've done a BLAST search or a diamond search or whatever, and it hits in your database this protein A, right? And so you assign the function of protein A to protein B. Next, or maybe in another data set, you have protein C, you do a BLAST against this database that also includes protein A and B. It hits protein B, and so it gets the same function as protein B, which has the same function as protein A. And then another search later on might be protein D. That has a hit to protein C, but you can see that it's going to receive the same annotation as protein C, but it doesn't have any overlap with the original functioning protein, okay? And so somehow this annotation from protein A has started to snowball throughout the ecosystem of all of these proteins, and it's because of these kind of matches that aren't necessarily informative. So we just need to be aware that these kind of things can happen if we're relying on these kind of sequence similarity tools. And then choice of database. So to go back to this question, the choice of database. So who is familiar with this cancer microbiome controversy? Not really controversy, I guess. Yes. Okay. Well, three people in the room. Okay. All right. Not good. No, no. So every so often in our fields, we always have somebody go a little bit rogue, do something really bad, and it kind of brings everyone down a little bit. So in 2020, there was this concept of this cancer microbiome. So what had happened is that Rob Knight had gone back to all the databases that had generated genome sequence data, and he was interested in seeing, are there bacteria associated with all these different types of cancer? And so he did all these sequence similarity searches, and he identified these kind of signatures of bacteria associated with each of these different types of bacteria. So that's great. So this has led to, it's been cited hundreds of times. It's provided data for 10 other studies that were published. It was very big news at the time. It sparked a big company. I think it got $250 million to start this company looking at using these kind of signatures of bacteria to help diagnose kind of cancers and so forth, and it looked incredibly promising. However, it turns out it was very, very flawed. So in 2023, there was a new study, which was from Steven Salzberg's group out of Johns Hopkins. And he basically found that a lot of the matches to bacteria were actually human matches. And so there's a big flaw in his search, in the original search when Rob Knight was doing his searches. He had a database of bacteria. And so if you're finding all of these genes and you're sending your MAGs or your metagenomic sequences against this database of bacteria, you are going to get matches to bacteria. Are they real? Probably not. What he hadn't done was included the human genome at the same time. And so when you do that, rather than getting, what's that, 500,000 reads which map to Streptococcus, you get 40 reads that are mapped to Streptococcus. Okay, so this was a huge error. I think last year, his group doubled down on this. They published another paper justifying why there was still a cancer microbiome. And it was very long, and there was an awful lot of supplemental material, and it didn't say anything new, but I think the idea was to try and overwhelm the reader to convince them to give up and say, okay, we give in. However, $250 million of investment will only get you, so far, and the investors kind of started pulling money, and it's turned out to be a pretty bad thing. So there should have been, when you're looking at your data sets, you should probably do these kind of sanity checks, right? So one of the cancers he was looking at were the glioblastomas, which are found in the brain. And I think there were deep sea microbes that were associated with these glioblastomas that they found. It's like, really? Shouldn't we be double checking these kind of results and making sure that what we've done is actually correct or not? Yeah.
What about these other bars? There's some other differences.
There are, but if you look at the scale, the one on the left-hand side is like hundreds of thousands. The one on the right-hand side is about 100 or whatever, right? So it's all just spurious matches. Yeah. So always be very skeptical about the matches that you get back. Do these sanity checks. And I keep reminding my students, do these sanity checks. Does this make sense? One time, and we processed one metatranscriptomic data set, and we processed it 7 times because I kept on coming across different errors. Like at one point, there's this deep sea virus, and it's kind of like, this seems really interesting. And we recovered the entire virus, and then a little bit more digging, it's kind of like, oh, that's a Phi-X that hadn't been removed properly, right? So it's these kind of things that we just need to be aware of. Yeah.
Well, just to further reinforce, I mean, we talked about this yesterday as well, and I just can't help myself but say it again, how the data management plan, that would, if they had a data management plan, someone probably would have said, hey, you should probably have the human genome in that as well. And then also always think of that data hierarchy. When you're looking at your data, but just the data that we created, like metagenomes and other things and other isolates and things, that has a hierarchy, but those databases also have hierarchies of how confident are they? Were they just confirmed via computation, or were they actually curated by an expert in the field? So always think about that. What kind of database is this? Is this a high curation level, or is this a low curation?
Yeah. On the plus side, it seems like there's a lot of people out there that are willing to throw money at you if you want to start a company. Did it get retracted finally? Okay, and what about the 10 subsequent studies? Okay, interesting. So it got
at the same time they were trying to think of the paper source they might still works then.
Makes mistakes, but this looks a mess.
Okay, good. All right. Anyway, that's a lesson to us all. All right.
That's a yes,
So we've got our list of genes, right? We've found our gene models. We've annotated them by doing whatever kind of annotation pipeline. So we've been doing this through BACTA today. And we end up with a list of genes associated with our MAG, and that can be a little bit intimidating. So if we think about these metagenomics data sets, if you start with 40 million reads or 80 million reads, or some people doing 200 million reads, that's a lot of genes that you've got. So it's hundreds of thousands of genes, and it's a list, and it's like, oh, well, now what? And so what you can think of doing is summarizing these into some sort of functional categories. So COG is probably the best well-known one, and people still use it for some reason. I don't think it's changed since 2003. It's pretty limited in its categories. So this on the right-hand side is actually, it's not from a microbiome, it's actually from gonorrhea. And the colors are just representing those genes that we don't know about versus those that we do know about, and what kind of functional categories they actually belong to. So there was, what is it? There's about 400 or so that are previously unknown genes that we've been able to reannotate and put into some of these other COG categories. But you can see the challenge here. It's kind of like these groups do not seem to be that informative. So if you see an increase or a decrease in one of these groups, what is that really telling you? It's kind of like, it seems really unsatisfying. And it's the same with gene ontology as well. Gene ontology for bacteria, it's improving, but gene ontology was originally developed for eukaryotic model organisms, and that's where most of the focus has been. Does anybody have any comments about their kind of experiences with gene ontology for bacteria, for microbiomes? No. Wow. Okay. What do people use for their functional categories here? KEGG modules, Tony?
I try to use Metasite as well to more organized, but Metasite is a big thing.
Metasite is interesting. Their pathways are relatively small, right? The nice thing about KEGG is the pathways are organized into groups of, say, 20, 50 genes, which is nice from a gene set enrichment grouping kind of point of view, because you can get a lot of genes associated with one particular functional activity. Again, the problem with KEGG is that the annotation was based on, I think, E. coli and also based on human to actually assign those pathways. So they may not be necessarily representative of what's happening in other bacteria. So again, just be aware of that. Yeah.
I've attempted pie crust back in the day.
Oh, nice. Okay. So Morgan, do you want to say something about pie crust?
Yeah, I thought pie crust is a way to extract or get functional systems data, right? So pie crust can put also different types of functional categories. And
So what are the pie crust functional categories based on?
annotation of genome. Do you mean what functional category?
Yeah.
Yeah, no, we have multiple. So we have K, and we have EC numbers, which mean that, right?
Yeah.
We have big now and KC.
Yeah.
And then most recent version.
Okay.
But the EC number is not the other side, okay.
Yeah. Yeah, so I think there's a real need for the community to develop a better kind of classification or gene ontology system for bacteria. Robin?
Genes already done.
You can above it, but then it's really fuzzy as to whether they really want to use it.
Yeah, like they're not gonna date it. And do it.
Yeah, but then when I look at it, it's like, but we kind of want you to pay.
I thought it was free for academia. Okay. Okay.
Like you can't get access to the pay.
Okay. So huge gap, right, in microbiome research is that we don't have a good kind of set of reference databases which assign things into functional categories, groups of genes that are doing the same kind of functions that will help us interpret these kind of data sets. So there's a huge opportunity, I think, for someone to make, maybe it's a community-based effort that we need to come together to do something like this, but it is a problem. Okay, so one way that we've tried to get around this problem, and I mentioned this a few slides earlier, is we've been focusing on specific subsystems. So rather than focusing on trying to assign every gene into one of these broad functional categories, what we do is instead, when we're doing our blast searches or we're doing our annotations, we might try and annotate different types of functions that have previously been assembled by specialists in these particular subfunctions. So for example, there's this tool called FeGene, which can predict ion capture and storage genes, and we've used that. For AMR genes, there's the CARD database, IGI tool, which can predict genes that are associated with AMR, and there's another one called, I think, is it ADRG or something? Anyway, we'll get to that. And then another thing that we've tried is exploiting maps of protein-protein interactions. So protein-protein interactions for bacteria, this gives you, these maps tell you which proteins are physically interacting with other proteins, and so you can get a concept of complexes or pathways of related genes based on these kind of physical interactions that they have with each other. So these kind of protein-protein interaction data sets have been generated for E. coli. There's a pretty good one for E. coli. For other bacteria, not so much. So we're currently working with a colleague, Andrew Amelie, to do one for gonorrhea. So these are really, really useful resources that we can start thinking about leveraging for layering on our metagenomic data. So on the right-hand side, these are genes that are involved in cell wall biogenesis, for example, that we've identified through these protein-protein interaction maps. And then what we're showing is these pie charts are representing genes that are expressed by different classes of bacteria under a certain condition. I think this was from maybe it's a chicken or a mouse kind of microbiome. So one example of these tools is PathoFact. So PathoFact, it was published, when was this published? 2021. And it predicts virulence factors, it predicts AMR genes. So this is a tool specialized for predicting a certain subset of genes within your MAGs. One thing to be aware of with these tools is when were they last updated, because you could be, unless you're kind of reading up on when that was published and seeing when it was last updated, some of these may just have been generated, say, in 2016, and it hasn't been updated since. And so some of these tools can get out of date. Unfortunately, in terms of grant funding, it's not easy to get grant funding to continue databases and database development. So just be aware that when you're using these kind of tools, when were they last updated? Maybe that speaks with the data management plan. So here's an example from Robin and Morgan, where they use PathoFact to show that the plastic sphere is actually rich in these antibiotic resistance genes and virulence factors. So each of these rows indicates a different MAG, and then the first set of bars are the incidence of those MAGs across different data sets. Then there's a number of virulence factors. It predicts a lot of virulence factors, I have to say. So probably about a quarter of the MAGs genes are associated with virulence factors, which is, I guess you take that with a bit of a pinch of salt. And then you get a profile of the AMR, different types of AMR genes associated with each of those MAGs as well. So using these kind of tools can give you some information. In this case, the authors were suggesting that there's an increased health risk, or there may be an increased health risk associated with plastic pollution. No, okay, there isn't. Okay, that's okay. Oh, there's not good evidence for that now. Yeah. Yes. Okay. All right. All right. So, okay, good. Good to have that clarified. But these kind of tools are very useful in order to focus in on different subsystems, and then you can interpret the data as you see fit, make hypotheses, and then maybe in future studies show that those hypotheses may be true or false. Great. Here's another tool, FeGene, predicts iron-related genes. So it combines blast searches and hidden Markov models. So again, you would get your FASTA file of all of your protein sequences that you have annotated from your MAG, and then you feed it into these kind of tools. In this case, FeGene, you download it. It's relatively straightforward to run. And it will output all of these ion acquisition, storage, oxidation, reduction genes. This was last updated in 2022, so we think it's still relevant. We used it to look at interactions between our favorite pathobiont, which is Trichomonas musculus. So Trichomonas musculus, it's kind of related to Trichomonas vaginalis, which is a pathogen of the genital tract. Trichomonas musculus infects the mouse gut. It's very prevalent. Who's doing mouse studies? Is anyone doing mouse studies here? Wow, interesting. So Trichomonas musculus, very prevalent in mice, and it seems to alter responses to bacterial pathogens. So there's an interaction between the protist, the host immune system, and then the microbiome. And it's been shown that if you're running trials, for example, of salmonella infection, if your mouse is infected with TMU, it's going to completely skew your results. So it's almost like a mycoplasma. Right? Remember when we were doing these studies in human cell lines, mycoplasma contamination was a big thing. If you have mycoplasma in your cell lines, it's going to screw up your results. In the same way, the presence of Trichomonas, nobody monitors for its presence, but mice generally, in a mouse facility, anything from 10% to 60% of the mice can be infected with Trichomonas. And if you're not measuring whether it's present or not, that's going to potentially screw up your results, especially if you're interested in studies involved in the immune system. Here, what we've done is we did a metatranscriptomics analysis, and we used FEGene to show that in the presence of this protist, it upregulates a lot of its own kind of proteins that are using iron. So it's sucking a lot of iron, which is normally a limiting kind of nutrient in the gut. And in response, the bacteria also upregulating a lot of their iron uptake systems. So there's this sense of this kind of competition for iron. Once this bug is coming in, maybe the bacteria are responding by upregulating all of this iron machinery to prevent the protist from getting the iron it needs to establish itself and colonize, right? So using these kind of subsystems, you're now starting to get some kind of mechanistic insights as to what might be happening in your microbiome. So in this case, FEGene was a pretty useful tool for us to understand that. But probably most of the focus and something that we're most familiar with is metabolism. Metabolism is, as I mentioned, anything from 1/4 to 1/3 of an organism's genes are associated with metabolic enzymes. Very informative. You can look at which different enzymes, which different pathways up or down regulated according to different sets of perturbations, what reliances are on different carbon sources and so forth. So there's obviously been a lot of focus on metabolism. There's a lot of tools out there which enable you to annotate enzymes to your mags. So we did one called Architect, which is a little bit complicated, and I'm not suggesting people use that anymore. DPC Transform is probably the winner these days in terms of accuracy and getting the widest coverage of enzymes associated with your mags. That will just predict which ECs, which enzymes are in your particular mags. But then one thing that Architect does, and another tool called GapSeq, and GapSeq is going to be part of the lab later on, is they will go beyond just predicting the ECs, and they will do something called metabolic reconstructions. So the idea is that you have your mag, you predict all of the ECs associated with the mag, you assign those ECs to reactions, and then you can start building these pathways. Within these pathways, you'll find gaps. So these gaps will represent enzymes that you couldn't detect. So maybe it's an analog. There's a protein which has no sequence similarity to anything in the EC databases. Maybe it's a spontaneous reaction, or maybe it's just a really distant author that you just couldn't detect, or maybe it just wasn't captured in your mag because your mag is in incomplete coverage. And so what these tools like Architect and GapSeq do is they fill in those holes, and they assume that in order for this pathway to make sense, you have to have these reactions, and we fill in these gaps to make sure that we have a complete pathway. And so one thing you can do with these metabolic reconstructions, you can generate them for each of these different mags. So now you have a set of metabolic reconstructions which describes the metabolism associated with each of your mags. And I'll mention in a bit how we've been using that for doing modeling. And this is kind of, pushing the boundaries of what we can do with these data sets. But I think from a forward-looking perspective, these kinds of approaches are getting a little bit more mainstream. Just as an example, with Doug Corver at the University of Alberta, we ran a chicken trial where we looked at the role of these antibiotic growth promotants on chickens. And we ran these metabolism detection tools. And what we did is we found that, for example, in this particular case, this is glycolysis, TCA cycle, pentose phosphate cycle. In response to the addition of these antibiotic growth promoters, we saw an increase in the production of these enzymes involved in energy production, right? And so there's this sense that you add these antibiotics to livestock feed, they're going to respond by upregulating a lot of their energy in order to respond to these antibiotic growth promotants. So this gives us, again, you're starting to get from these kind of functions, some sort of mechanistic insights as to how your microbiome is actually responding to the environment. In a more recent study, this was with Ben Willing, who's also at the University of Alberta, we looked at these metabolic differences between poultry gut microbiomes, ones that had Bacteroides, ones that didn't have Bacteroides. And we generated 237 mags from 33 samples, and we can look at all of the ECs that are present in each of the mags. And so each of these different mags each are contributing different sets of enzymes within the community. And what was interesting is that we found that those communities that are coming from the Bacteroides, that have Bacteroides in them, their metabolism seems to be very different. The community metabolism seems to be very different to those that have that didn't have Bacteroides. So Bacteroides is somehow promoting a very distinct kind of metabolic universe, if you like, within that gut of those chickens when it's present compared with when it's absent. And so to investigate this a bit further, we can turn to something like metabolic modeling. We have these metabolic reconstructions. Metabolic modeling, there's a number of tools that are out there now, enable you to start going in and seeing what is actually happening within these communities. And so there's a number of tools out there. Probably the best one, the easiest one, or one that's relatively easy to use, is this Bacterina. And this was published back in 2017. And we're starting to see some of these publications now starting to come through, looking at different microbiomes, predicting metabolism, metabolic interactions within these communities. So the idea is you get your set of metabolic reconstructions associated with your mags, associated with your community. And then you put them on this two-dimensional grid. That's this thing on the right-hand side. And then there's a whole set of black box magic. It's called flux balance analysis. You don't need to get into the details, but it's a mathematical formulation which effectively tries to maximize the growth of each of these different bacteria by looking at the flux and changes in flux associated with all of the enzymes and the metabolites that you feed into it. So you also have to feed a diet into these kind of systems. And then on the right-hand side, we've got this community. Each of these dots represents an individual bacteria. And then as you're running the simulation, these bacteria grow at different rates in response to these predictive models. So we've applied this to our chickens, and on the left-hand side is my AI slop picture of Chickmatron, which is what I've come up with. And when we applied it to our poultry gut models, it does a pretty good job of predicting the production of short-chain fatty acids. So the one it wasn't so good at was the prediction of butyrate in these communities that lack Bacteroides. But then we looked at that, and it should actually be doing a better job, or rather, we don't know what's happening with butyrate. Our model is predicting that some of the bacteria that are present in the communities that didn't have Bacteroides are actually producing a lot of butyrate, but that doesn't seem to be happening when you're measuring experimentally. So maybe there's, in the gut, maybe there's some absorption or something like that going on. These things can provide these hideously complex kind of diagrams as well. So the Sankey diagram, what we're showing here is really when you have Bacteroides in yellow, that's responsible for the production of a lot of metabolites. However, when it disappears, you see that E. coli in green is now taking over a lot of its kind of responsibilities for the production of those metabolites, along with a couple of other taxa as well. So you can start seeing what happens when you remove some of these keystone taxa, who's taking up the slack, who's doing these kind of productions. And we see a complete kind of reorganization. So in the left-hand side of this heat map, we're showing metabolic interactions. So the metabolic interactions is who is providing metabolites from one taxon to another taxon. And the key thing to take here is on the right-hand side, when you don't have Bacteroides, you get like these blocks, right? You see these square blocks. These represent subcommunities within the entire poultry gut communities, where there's 4 or 5 different taxa, which are all kind of sharing or contributing to each other's metabolism. So again, this kind of reorganization within these kind of communities in terms of metabolism. So this is a lot to take away, but I just wanted to put this out there as this is kind of, I think, a direction that we can start taking some of our analyses of these microbiomes and starting to look at, you know, what does it mean when you're getting up or down regulation of certain of these enzymes? What is the consequence in terms of the community from an ecological perspective? Can you start using these for predicting diets as well? So one thing that we've been looking into is screening lots of different diets and how does that alter the production of different metabolites. So short chain fatty acids, we think they're going to be beneficial for us. By adding in a different diet, using these simulations can re-predict diets that increase these kinds of metabolites. So these kind of tools, these are R packages. There's organization involved in putting things in the right kind of format or whatever. But again, these are not things that you should not, these are things that you shouldn't be, that you should be confident that you can run. I'll put it out there. So again, I think these are the kind of directions that we see potentially the field, particularly in terms of metabolic interactions within microbiomes, potentially going as well. And of course, one big caveat, these are models, and I can't remember who the person is, but the classic quote is, all models are wrong, but some are useful. And so what we're using these models for is really for doing predictions. They're hypothesis generating. You can go back to the experiment. Is my hypothesis correct or not? If it's not, go back, refine the model. But you're learning more and more about the ecology. You're learning more about the community. So we think that these kind of approaches are useful. Okay, any questions on that? We're not going to be doing any modeling in the lab, so don't worry about that. I'm just putting that out there as a kind of a future-looking thing. Yes, Morgan.
Okay, there. Metabolic prediction.
Yes.
If you just look at, you know, the genes that are involved in
Mm-hmm.
advanced heat flux analysis, what does that look like? Is it different, like based on the presence of the genes, or is that really an emergent once you do that?
Good question. I think it's only emergent when you do the actual modeling, because it will depend on the diet that you're feeding in as well, right? And then so these metabolic reconstructions, you basically got 1,000 reactions which are connecting this huge set of plumbing, right? And you're turning on taps at different ends, and then you're seeing what's flowing through these ends down here. And there's a lot of these non-intuitive, non-linear kind of relationships in terms of having the presence or absence of these different pathways that are contributing to the overall flux. And so we will see that some of these organisms will have the enzymes associated with with the production of butyrate, for example. But in terms of quantifying the use and the flux that goes through those different enzymes is not going to be seen just by looking at the abundance of those enzymes in different bags. You actually do have to do the simulations to see substrates that might go through those pathways. Do they get redirected somewhere else to contribute to the growth of other taxa, for example? So I think this modeling, but it's a good point. So I do get your point that we could look at that. That would make a suggestion as to whether it is or not, and then using these kind of tools to actually demonstrate this. Is that a long-winded answer? Is that a satisfying answer for you, Morgan?
That's fine. More general.
You see, you come to my lecture once, and now you want all these questions.
Thanks.
All right. Okay. Yeah.
No, I just want to comment about, studies about, they annotate their genes and their metagene for May. What's a reasonable amount of genes that they could put a function on? What's the expectation there? Like if they, if they annotate 25% of the genome, I feel like that's okay. That reasonable. Just wondering if you could throw that out there for the audience, or if anyone else has suggestions.
No.
It's like an expectation.
Follow-up, what if it's in a non-model system?
Yes. So yes and yes. So I think maybe this was mentioned yesterday as well, that if these are non-model systems, then you're obviously going to get a lot less. From our metatranscriptomics, we do a lot of metatranscriptomics. Probably in 2016, we were able to annotate 20% of our genes with putative functions. Now we're getting up to about 50 to 60% of putative functions. And this is coming from human data sets. But if we move away to something like a deep sea data set, then that plummets our ability to do the annotations. So the annotations absolutely depend on how well characterized your genomes are. And maybe Steve will have a comment on this as well.
Yeah, it depends on the system, obviously, like, if you're in a non-model system that's using a different codon usage table or something like that. I don't know what to say. That's a hard problem. But for MAG, to kind of go back to Morgan, I mean, typically we can only annotate between 40 and 60% based on the reference databases that we have in general, whether it's a reference genome from an isolate or a MAG. And the problem's compounded in the protein structure databases too, because a lot of the current solutions are based on predictions, right? So you've got a model based on a model, as Ryan pointed out, those become simulacra of increasing non-reality. And so I think just to kind of go back to the nature of the problem, very few annotations are functionally validated. Yeah, so even if you get a hit to something in the TCA cycle, that you're pretty confident that that's the trait lysines for a reverse TCA cycle as an indicator. It's not validated unless somebody goes in and looks at it biochemically. And so almost every one of these models that we're producing is really quite a fiction. So it's really important to be careful about over-interpreting. And the real problem with MAGs is that very few people report the information loss in their process. And so, like, if I start off with 1 billion reads, and then I publish a paper on 237 MAGs, that may only be 10% of the reads that I started with. So it's a subset of the community that I'm actually describing now when I do these calculations.
Just the figure that Brian showed us.
Yeah, it's just really important to point that out. Like if I want to look at microbial community interactions using MAGs, I may have discarded 80% of the data of my community. What am I looking at? Again, it's a fiction, and it could be a hypothesis building experience, but it's not necessarily representative of reality. So this is the grand challenge. How do we get truthier? I don't know the answer to that, but probably working with simpler communities would be a start. But MAGs are very dangerous, right? Because you have a fragment, you have an incomplete gene. You don't have a closed reference genome most of the time. You have an incomplete representation. It's not an isolate. It's a population level construct. It does not exist in nature. And now we are trying to attribute metabolic inference onto that construct. And then it also represents a subset of the community. No. And then we build all these elaborate stories around that without recognizing that maybe not even the important member of the community. So I would just be very cautious. And if you're looking for, to go with the all models are bond with some are useful boxes, the modeler box. One might also come up with the absence of evidence is not evidence for absence. And so many papers look at mags for the presence of genes. And they compare, oh, look, we found in these mags that have nitrate reductase, therefore these must be involved in denitrification. But the problem is then you don't get the reciprocal. I'm missing a lot of nitrate reductases as well because the mags are incomplete. So I don't know if you guys wanted to comment on that, but good. Okay.
I would also say though, I think for enzymes, I think certain classes of proteins are much harder to predict than others. I think enzymes, because we've been working with enzymes for 150 years, they have been very well characterized. We feel pretty confident about the predictions associated with a lot of these enzymes. That's why something like DPC transformer, for example, we think is a pretty accurate tool in terms of doing these kind of annotations. The other thing, back to Morgan's point with the lack of annotations. So this was an exercise that we did with gonorrhea. So even model organisms where we do have a complete genome, would there, in this case, it's what about a third to gonorrhea has 2,000 genes and 800 of them were previously uncharacterized. So that gray bar at the top, those were genes that we could now annotate after performing structural modeling. So using AlphaFold. So AlphaFold is potentially a new method that we can start thinking about in terms of addressing this gap between the 40 or 50% of the genes that we don't have any annotation for, and maybe using these kind of tools like AlphaFold to make these kind of predictions. Okay, was there a final comment over there? No?
Sure, one last one. What do you think about Human and the annotated functions? And
Sorry, in terms of
Human on, say, directly annotating functions directly on the reads and not mags. That approach. We wouldn't talk about that. Curious about your opinion.
my opinion is that I don't like human 3, or is it 4 now? I don't know. We ignore it because it gives you what it thinks you should get. And that I feel insulted when a tool tries to do that for me, because I want to know what's happening under the hood. There's output that we need that you can't get out from the human 3 output in terms of trying to get abundance data or different types of data that is kind of buried deep within the algorithm. So we don't use human 3. And I think why not assemble? Well, with metatranscriptomics, that's effectively what we're doing. We're annotating to these short sections of sequences, and we're using that information to inform on the functions associated, encoded, expressed within our communities. And that's what I'm going to get on to next. So I'm not adverse to that kind of approach in general, but the way human 3 does it, I think is a little bit buried. So we have our own pipeline for annotating for metatranscriptomic short reads as well. And again, buyer beware, that's why you need to do these sanity checks to see whether these annotations really are making sense in the context of your community. And there is a comment here.
Or what do you think?
It's great to run, change to run. From a practical standpoint, it also, I don't really know how I feel about it, kind of assumption. I think partly because I have issues with native first text authentication, and it does that as a first pass. Yeah, so, but then I prefer to just make a database myself and that something, so I know exactly what I'm doing.
I mean,
it's
we
necessarily
use
a
it in the
problem.
past a lot,
Yeah.
I don't think it's the worst. Well, I think it depends on, I think for obviously well understood human, things like that. And it's sort of designed to pass it on, I think it's an okay approach for me. Yeah, I hear what you mean about not being a big internal person. But it's I think it's a throw away for sure. But it's fun. But yeah, we also tend to really do it ourselves. But we've used it obviously as a tool, as a first pass to find out what we're doing. In the bomb.
It is 10 o'clock, so I'm going to rush through.
Yeah,
What's that?
go ahead, John.
All right. Okay, so last section, I just want to try and convince some of you to start thinking about maybe metatranscriptomics as a way of informing a bit more about our microbiomes. So as you're probably familiar, metatranscriptomics, rather than doing DNA, sequencing DNA, you're doing all of the RNA within a sample. So the idea is that this can actually reveal active functions as well as the taxa that are responsible within a particular sample. Here we've just got an example from these are cell wall biogenesis genes, some of which are being upregulated in the presence of antibiotics. So this is from a poultry gut. So this is a way of showing in response to the presence of these antibiotic growth promotants, the bacteria are increasing a lot of the genes involved in the production of cell wall. Makes sense because the target of these antibiotics are the cell wall. So that's great. Again, it starts getting into mechanistic insights into the microbiome. So one of the earliest studies that we did for metatranscriptomics is from 2017. Very simple study, 4 mice in each group. We fed them 2 different diets. High-fat diet, low-fat diet. And then we had wild-type mice, and we compared them with this PLIN2 knockout mouse strain. PLIN2, perilipin-2, it's a gene that the mouse expresses. It interacts with lipid droplets, helps with the absorption of lipids, regulates lipid uptake. And so if you delete this PLIN2 gene in mice, it abrogates some of the bad effects of a high-fat diet. So they're not able to uptake so much of the lipids. So we were interested in what impact does this knockout have on the gut microbiome? Relatively simple question. We did the analysis of taxa, and we found that there is no significant deviation in terms of which taxa are active within these samples or not. So under a high-fat diet, under a low-fat diet, PLIN2 knockout mice, the gut microbiome seems to be very similar to the wild-type microbiome, at least in terms of the taxa that are present. However, when we look at the expression, we found that there are over 1,000 highly expressed microbial genes that were significantly showed significant differential expression. Many of these were associated with amino acid metabolism, energy metabolism. And so here is a section from glycolysis, and within that box, all of those kind of dark red blobs is an enzyme that is downregulated in PLIN2 knockout mice. So what seems to be happening is that PLIN2, the microbiome of these PLIN2 knockout mice are actually downregulating this section of glycolysis. And so this gives us this kind of model where in wild-type mice, you feed it a high-fat diet, you have a lot of triglycerides, PLIN2 is present, you can absorb a lot of these triglycerides into these lipid droplets, and so you just get normal energy metabolism for the microbiome. However, under the PLIN2 knockout mice, you now have way more fat and triglycerides in the lumen of the intestine. It's not being absorbed by the host. So as a consequence, you've got all of this extra kind of fat that these microbes can now use for growth. And so now it has more than sufficient lipids for production of energy. So it's down-regulating those kind of pathways and then converting the lipids instead, shunting them through different pathways. So again, if we just use 16S, if we just use metagenomics, we wouldn't get these kind of insights. It's only through doing metatranscriptomics that we get a sense of what is actually happening, these kind of mechanistic effects. Now, there are some issues with metatranscriptomics. Sample collection is one. RNA degrades very easily. Ideally, what you want to do is, as soon as you've got your sample, place it on -80 as quickly as possible. We have trials with kids with IBD. So they have to produce samples at home, and so we send them home with these kits. This OmniGene gut kit, which came out in 2022, seems to do a reasonable job. So there are these kits that help stabilize these RNAs. Another problem we have with metatranscriptomics, there's a lot of ribosomal RNAs, right? So if you just sequence all of the RNA associated with your sample, around about 95, 98% of it is just going to be ribosomal RNA reads. These aren't that informative. And so there are these depletion kits. So this Ribo0 kit that can deplete the ribosomal RNAs. And so you're boosting the quantity of the messenger RNAs that you get. However, these are taxonomically sensitive. On the right-hand side, these were parasite-infected mice, and we had a lot of ribosomal RNAs despite this depletion kit, because this depletion kit did not deplete the parasite ribosomal RNAs. And so a lot of our signal was ribosomal RNAs from the parasite. Another thing, this was brought up, I think yesterday, maybe in the beginner's workshop, is abundance. So relative abundance versus absolute abundance. So there are different ways that you can quantify absolute abundance. We do a spike-in. So there are these spiking kits. In this case, it's this Zymo kit, and it's got these 2 bacteria that are never normally found in any microbiomes. You spike those in, and you can use those to normalize your expression. And without normalization, you can end up with very misleading false positive results. We have a pipeline for doing metatranscriptomics, and I would be remiss if I did not advertise this. It is undergoing a re-updating. So again, just to mention that it's important to ensure your tools are updated. So our databases were from 2020, so we're now updating everything to be more up to date. Another challenge with metatranscriptomics is this concept of normalizing expression through these RPKM. This is a concept from RNA-seq. So if you have a longer gene, you're going to get more reads associated with it. So you have to normalize for the fact that if you have long genes, you're going to get more reads associated with that, and you're just normalizing for the length of the gene. And then finally, what does the data look like? So at the top, these are 16S profiles. In the bottom, metatranscriptomic profiles. These are from Chicken Seeker. They look pretty similar. However, you can see that the purple taxa don't seem to be very abundant. So from the 16S analyses, they don't seem to be very abundant, but from the metatranscriptomic, they seem to be very active. And then you've got the yellow and the green taxa at the bottom in the seeker. They tend to be quite abundant, but they don't seem to be very active. Okay, so you're starting to see that metatranscriptomics are really informing on who are the important members within your communities. Very excited about this initial data set from Pakistan. These are 23 stool samples. So we have got first trimester and third trimester samples, and we're seeing the sort of things that we would expect to see. So there's majority are coming from Firmicutes bacteroides, as we might expect. Bifidobacteria appear to be increasing the third trimester. Again, this is what we might expect. And then one sample in yellow that I've highlighted with the star there, that shows evidence of a Giardia infection. So this is exciting for us because here is one of these pathobionts that we're interested in. This mother seems to be having an episode of Giardia infection. We've got some medical records to see if she was having a diarrheal episode at that time. And we also find viruses as well, enteroviruses and even plant viruses that might get back to this concept that we're detecting these kind of dietary components within these samples as well. So this is great. Metatranscriptomics seems to be informative, seems to be capturing exactly what we would expect to see. And so we think metatranscriptomics is an informative technology that we would encourage people to consider. There's a question over there. Yes.
to some people at the University of Toronto, and they had urged us to do proteomics instead of metatranscriptomics, because they said it was easier and we didn't have like the issue with the RNA being unstable. I'm just curious of your thoughts on the like protein versus transcripts, although I kind of know.
Yeah, I guess I've been disappointed by some of the metaproteomics. Maybe it's changed in the last couple of years or so, but just the number of proteins that they're able to identify don't seem to be very comprehensive. So some of the data sets, or at least from about 5 years ago, was about 400 proteins, right? With the metatranscriptomics, we're getting 500,000 kind of different transcripts. And it depends on the quality of the databases that they're then searching against as well. I don't know about the cost of the proteomics as well. I know that, so there's also different types of proteomics as well. So one way of using proteomics is just to see who's there, as opposed to measuring all of the genes that are actually present as well. So there's also different aspects associated with that.
Or like what
Yeah. Okay, yeah. So you really were trying to go for these sets of genes, and how successful was that? Okay, how many proteins did you find?
I don't.
Okay, yeah.
I've only done proteomics on single cultures, single bacteria cultures, but it's kind of difficult because you have to extract the cellular, extracellular, and potentially membrane-bound separately. You're kind of looking at different aspects than you would be with something else. So you can kind of try and do everything, but also it's really difficult to map proteins back to a single genome when you know the genome, let alone in meta.
I mean, false discovery is a big problem with proteome, both from the peptide matching and for the mapping problem itself.
Yeah.
Well, what we found is that, I mean, there's nothing wrong using proteomics as a validation. Like, if you really want to know if gene X is manufactured by the cell, having a protein diagnostic is great, especially if they're hypothetical protein. And you're trying to link them to some kind of interesting process because they seem to be enriched under one set of conditions. But the transcriptomics tend to be pretty robust, actually. And also a great way to validate gene models. Because if you predicted it using prodigal and the transcripts are there, you can be more confident that prodigal got it right.
Yeah, absolutely. Yeah, so that's a great point. There's the different fractions. You've got soluble versus membrane proteins, right? And you're not going to get as comprehensive a set. The databases also with the false positives, because you're working at the peptide level, not at the nucleotide level. And so the identification of the taxa is going to be that much more trickier. But on the other hand, I wonder, it also depends, do you care? Do you care which taxon is providing that protein or not, right? So there is that, right? And then finally, this was a study. When was this one from? This one was 2022, and it compared metagenomic data with metatranscriptomic data. And they looked at these data sets over time, and they found that the metatranscriptomic data, which looks at the expression associated with different taxa, was actually a better predictor because it was looking at activity of each of these different taxa over time, and it was a better predictor of what the community would look like in the future. So again, another kind of takeaway from metatranscriptomics versus metagenomics. Now, ideally, what you'd want to do is metagenomics and metatranscriptomics, but that's really expensive. So people don't tend to do that. But if we had money, and potentially metaproteomics. Anyway, I have gone way over time, so I do apologize for that. Were there any final comments?